Title: CRISPR, clustered regularly interspaced, short palindromic repeats. An excerpt from a conversation with Eugene Koonin, PhD, November 2022. Patricia Flatley Brennan, RN PhD, Director, National Library of Medicine. She is seated with Dr. Koonin at a table in a large room.
I'd like you to reflect for a moment on the contribution that you delight in the most in your scientific career.
Eugene Koonin, PhD, NIH Distinguished Investigator, NLM NCBI.
Perhaps I should mention the CRISPR system.
Yes.
I will not explain that acronym. It gets a bit hairy.
That's fine.
But what, on the other hand, what CRISPR is widely known, because this is known as the new methodology for genome engineering and editing, which is far more orders of magnitude more efficient and fast than previously available ones. But in biology itself, this is something also of great interest, because CRISPR is an adaptive immunity system in microbes, in bacteria. And before this was discovered, it was no one even suspected that adaptive immunity, fundamentally similar to our own, existed in bacteria. And with my colleagues, through comparative genomics approaches, as usual, we have been instrumental into the discovery and further characterization of this system. Many labs contributed, but I think our contribution is noticeable, and has been so for quite a few years now. I guess if I need to mention one achievement, that would be that.
I think that you've helped me understand the range of new proteins that you're discovering that have the features that are important for this approach to modification. Do you think about the long-term disruption of evolutionary pathways that CRISPR can cause?
Do you mean the practical application of CRISPR, or?
Well, I guess I'm asking from the perspective of if we're changing a structure through a biological intervention, are we expecting that change to be permanent and heritable through next generations of that entity?
There are different ways to make these changes, and
Okay.
they are controlled. When we speak about genome editing and genome engineering, by definition and by design, this implies heritable changes.
Okay.
And this is what we want. In case, let us say, of treating genetic diseases, you want to change the genome. Of course, the burden of responsibility is heavy in such cases, in the sense that you want to make this particular change, to revert this particular mutation that is responsible for this particular disease, rather than anything else in the genome. And this is probably the challenge of so-called off-target effects is probably the greatest challenge in the development of the CRISPR technology, or for that matter, any approach to genome modification. Of course, CRISPR has many other applications that allow us to manipulate processes in cells without changing the genome.
So there are more applications than gene editing.
There are far more applications than gene editing.
And those help in understanding basic biology, those help in creating novel environments?
These certainly help understanding basic biology, but they also help manipulating the functions of cells in a desirable way, and can even have therapeutic applications, just not heritable ones.
I see.
It can change transiently the way cancer cells function and contribute to therapy.
Text. CRISPR. Clustered regularly interspaced, short palindromic repeats. An excerpt from a conversation with Eugene Koonin, PhD, November 2022. Gray and blue logo. NIH, NLM, serving scientists and society.